Well, if you've survived this long, this is the last of these great lectures that I've given you. By now, you certainly understand cell structure and function. You certainly understand how a cell organizes itself to produce proteins, how it accumulates energy, how it goes about producing other cells, and of course, carries out the process of reproduction. You've also learned some of the key concepts of genetics. You understand also how DNA goes to RNA, RNA goes to protein. All of that encompasses the central dogma of biology per se. And we've also touched to some extent on some of the glue that holds biology together, that is evolution. And to some extent, maybe to a very little extent, we have touched on some basic concepts in ecology. So where is all this going? So what is the future in biological research, and what is the direction that it will probably take within your lifetime? We must put things in perspective. First of all, we have learned more in the area of biology in the last 50 years than we have in the entire history of man's accumulation of knowledge. In the next 50 years, we will open up new avenues, and it's difficult to say exactly where all this will end. So what I will do now is look at some future directions in biology. Now, one of the major areas in biology is biotechnology. This is a major tool that's moving biological discoveries. To define biotechnology, it is basically the manipulation of biological processes for medical, agricultural, and industrial objectives. Some examples of biotechnology would be transgenics. We'll discuss that in a moment. Recombinant DNA technology, cloning technology, some new avenues in forensic, and lastly, genetic analysis. Here we're looking at a tree that's glowing. Now, what has really happened here is that we have taken the genes that result in the firefly luminescence and transferred it into a plant. And we basically have produced the essence of a firefly into a plant. Kind of cool, actually. One day you'll walk down the road at night and you'll see trees that are glowing as a result of the chemistry that has been incorporated into these plants. Another interesting avenue of research using recombinant DNA technology is in the area of aquafarming. Here we're looking at two fish. Both are the same age, except the fish to the left obviously is much bigger. The fish to the left, we have incorporated specific genes that enhanced the growth characteristics. Here we are looking at an example of transgenics, or recombinant DNA technology. These goats, prior to birth, we incorporated specific human genes. So they are producing human proteins which will be used for therapeutic purposes. For example, growth hormones can be produced by these goats, perfectly compatible with human growth hormones. In addition, a whole spectrum of specific antibodies can be generated by these goats. Now we're looking at cloning technology, and we will discuss this in more detail in a moment. We're looking at two horses. The reality is the small horse is actually a clone of the big horse, and we'll show you how this process works. So let's look at DNA technology, recombinant DNA technology. Basically, we have to look at this process as mere cutting and pasting, as you would, for example, in a document on your software. Gene splicing What we're doing is we are cutting a piece of DNA from one organism and transplanting it into another organism. The key tools in gene splicing or recombinant DNA technology is, first of all, we need to identify the gene of interest. Secondly, we use molecular scissors, referred to as restriction enzymes. And thirdly, we find a way of taking that particular gene, incorporating it into another host. So let's see how this process works. We're looking, of course, at a piece of DNA, double-stranded DNA. And if we look carefully, the first thing we would do is target a specific gene, as the arrows indicate. If we add unique restriction enzymes, we are able to specifically cut a given gene from its parent genome. As you can see towards the bottom, we have cut the entire gene out. Now, once we have a gene from a given organism, from a human or some other organism, what we will now do is transfer it into another organism where it will take on the characteristics of normal genes and produce desired proteins. So how does this process work? First of all, we need to have a vector. A vector is nothing more than a vehicle for transferring a gene. We're looking at a bacterium, and the bacterium has two basic nucleic acids. One, it has the regular genome to your left, the bacterial chromosome, and to the right, it has plasmids, small circular DNA. A given bacterium will have thousands of plasmids. These plasmids replicate in the same way as the bacterial genome does. As you can see to the bottom, you see this nice little circular DNA molecules. These plasmids will be used as a vector for transferring genes from one organism to another organism. So let's put this whole process together in this model that we show you on the screen. Okay, to start with, let's pretend that we are going to produce insulin, which is, of course, a protein, a very important protein used for therapeutic purposes. Now, insulin is controlled by a gene. Of course, if you remember, genes produce proteins. Now, the first thing we have to do, as you can see to the left, we identify the gene that produces the human protein, in this case, insulin. We identify it, we target it, and we basically cut the gene out from a human cell. Now, once we cut the gene out from the human cell, as you can see, we're moving from left to right. We then take our plasmid, which is our vector, we open it up and insert the human gene as shown to you in dark blue. The human gene now becomes integrated into the plasmid. This is referred to as a recombinant DNA molecule. We then take this circular plasmid, put it back into the bacterium, and then take the bacterium and put it into a vessel and allow it to replicate. We know how fast bacteria replicate. Within a matter of a day or two, we'll have billions of microbes. As this bacteria replicates, the plasmid and the insulin DNA is also replicating. Now, once we have a vat full of bacteria, the DNA goes ahead and produces protein. In this case, the human gene, which is incorporated into the plasmid, is now producing proteins. As you can see, the green material that's being produced by the bacteria, that is a product of the recombinant DNA that we produced. In this case, we are making insulin. The insulin, of course, can be purified and used for therapeutic purposes. So, in this fashion, we can take genes, whatever genes we identified that we feel are important, move them from one organism to another organism, and this is the essence of recombinant DNA technology. And here we're looking, of course, at purified proteins that is produced by recombinant DNA technology. So now let's look at cloning. All of us realize that the genes for making a complete organism is present in every cell of that particular organism. Basically, your skin cells have all the genetic material to make another person just like yourself. Your muscle cells have all the genetic information for making another person just like you. They don't do that because these genes are locked in place and they do not express themselves. So let's see how cloning technology works. We take a sheep, we take some mammary cells and put them in culture, as you can see. We then take the black-faced sheep and pull out an egg. In this case, we take out the nucleus from this egg and discard it. We then take the cell from the white sheep that has the genetic component of the white sheep and we fuse it with the egg of the black sheep. Once we fuse this, the embryo, as you can see towards your right, is implanted into the uterus of the black-faced sheep, and after the appropriate gestation time, what do we get? We get a little sheep, which is Dolly, which was the first one done in this process, and this Dolly is identical to the white sheep that we started with. So what we have done is cloned the white sheep. Now, can we do this with humans? In theory, of course. Is it ethical? That's an issue that society will have to determine long term. And here, of course, we're looking at Dolly and its surrogate mother. One of the problems in doing this is that it took a lot of starts and stops before this process worked. So therefore, it would be unethical to try this with humans. Unfortunately, Dolly did not live very long. It developed some problems, and we're not quite sure if this was directly related to the cloning process. Another interesting and important avenue of research is in the area of forensic. All of us have heard of the use of DNA in convicting individuals and identifying individuals as being the culprit for a given crime, and so on. How does this process work? Every one of us, when we come to a given scene, leave behind some material that will have DNA in it. Now, normally, the DNA that we leave behind, whether it's in some cells that we shed or some hair follicles, normally there's not enough material to be able to do a complete genetic analysis. We all have similar DNA, but the differences are enough where we could differentiate without any doubt one individual from another individual by analyzing DNA. But with so little DNA left behind, how do we go about analyzing this minute sample? Well, in the 80s, a discovery took place in the area of PCR technology, polymerase chain reaction. In this process, what we're doing is we are taking the material that's left behind that is in minute quantities, let's call it the target DNA, and we amplify it. We're able to specifically amplify the target DNA. So let's look for a moment, starting off on the top. The double-stranded DNA that's left, let's say, at a crime scene, we are able to amplify that material. We do it by opening up the double strands, attaching specific primers that will specifically trigger the duplication of the molecules. So we go from one molecule to two double-stranded molecules. We'll repeat the process. We get 4 molecules, and we continue to do this process so that we end up with 10 billion molecules of DNA. We start with one from the original specimen, and through a series of amplifications, we now have a bucket full of DNA. Boy, that's great. So we have basically amplified the target. Now, once we amplify the target, the rest is relatively easy. We have so much DNA that we can now do a genetic analysis. The genetic analysis that we do following PCR technology is a system that allows us to separate the DNA that we get into small pieces and putting these pieces onto a gel and looking at the profile of the pieces that we get. As you can see, we're looking at a gel apparatus. We have 3 samples being put in place. Each sample being different will give a different profile on the gel analysis. As you can see, the profile shows you the bands that you get. No 2 profiles from individuals will be identical. So therefore, your profile, my profile will be unique. The chances of your profile being identical to my profile perhaps is 1 in 10 billion. Here we're looking at basically doing a DNA fingerprinting, which is basically doing a gel analysis or profile. As you can see, we're looking at blood that was taken from a victim and blood that was taken from a murderer's shirt. On the gel analysis, you can see where the fingers are pointing. The profile that's produced on the gel are identical from the victim and the murderer. So this is basically the process that we use PCR coupled with gel analysis to identify a given specimen belonging to a given individual. Another interesting area that we should be aware of in the future is the ability to put a complete laboratory-based analysis on a single chip. In this case, if we want to completely analyze an individual's genetic capabilities, let's say, or their potential for developing diseases, we have hundreds and thousands of markers that we would have to identify and try to determine whether an individual has or does not have a particular marker that would indicate a possibility of developing a given disease or whatever. Now, what we can do is we can develop chip-based analysis. As you can see here, we can actually do 10,000 points, which means 10,000 gene analysis on a given sample. So therefore, in the future, you may go to your doctor and get a complete profile of your genetic potential, your genetic risk to certain diseases, and other information that may or may not be useful. Okay, so where are some other interesting avenues of biological research? Within the next 50 years, we certainly will understand the mechanism of aging. And we certainly will be able to regenerate organs and tissue. Another important area that is ripe for research and that great strides are made is in the area of memory and how the mind works and how we're able to put together this vast amount of information and retrieve it at a given point. In addition, we will probably be able to resurrect extinct species and probably create new species. And finally, perhaps the most ambitious avenue of research, we will probably make significant headway to determine if we are alone or not in the universe. Thank you and have a nice vacation.